opium poppy genetics, and he wants to share his work with you today. So, take it away, Raj.
Thank you, Khalid. Good afternoon, everyone. Finally, we all can meet and, maybe fully clothed. We're not on a Zoom calls, right? I need to stand here.
Do you want a roaming one?
Yeah,
You can
that'd be great. Perfect. So the name of our company is Emerzine, and we are based in Dublin, California, East Bay. The whole team started out of my PhD, and I have a PhD in genetics from Australia, and the name of the company is Emerzine. This was 12 years in making. Right after my PhD, we went and applied for licensing to get a poppy license, right, to grow. It was a big hurdle, laws and regulations didn't permit us. Finally, we are here. So we are the world's first non-addictive pain relief medication. We are working towards it. Our leadership team consists of a diversified background with doctors, farmers, investors, and all of them have one vision. We want to help the humanity, because there is a lot of drug abuse and addiction that you can see in almost every single major cities, And this is, some of them are not aligned properly, but we have good industry affiliations. The main problem is opiate crisis, right? As I talked before, if you look at these figures, they are published by International Narcotic Control Bureau, which is an INCB, published by United Nations. So it's not like published by any country or something. It's this opiate crisis statistics are managed by a bureau based out of Vienna, and every year it's staggering, like what you see, and because of the pandemic and use of all these opiates increasing every year by at least 20% every year. And look at the deaths that's happening every year. And addiction is just not going away. This is the crisis right now. We just got off from the pandemic. And if you look at the statistics, 130 people die every day, and 81% of the overdose deaths are causing the homelessness in many major big cities. So our solution to this global problem, we don't want to say we are the complete solution, we want to be part of the solution. And we have a new variety of poppy, which is a perennial variety in agriculture terms. Annuals and perennials. Annuals you grow 1 year, perennials you grow multiple years. So we have a different strain of poppy that can be grown multiple years without replanting, and that has a unique alkaloid pathway which is free from morphine. And we are working with Merck, and we are trying to work on CRISPR technology and how we can edit the genes that are related to morphine, and we can edit the genes and also manipulate the enzymatic pathway. We have a solvent-free extraction process. We have partners in Europe who specialize in this. And we also think that our poppy has other alkaloids that are highly, highly effective in treating noscapine and atropine. Both our compounds are used as sedative for wild animals. If you go to the zoos, if they want to tranquilize some animals, atropine is the compound they use, and that is in shortage right now. But with us, we have that seed that we can produce atropine in a quantity that's substantial to the world. As I said before, 2 varieties of poppy are grown in the world. Hundreds of years, people have known only about somniferum, which is the traditional morphine variety. Annual and labor intensive because you need to take that crop every year and replant it every year. And our variety is perennial, and the yield of our variety is 5 times the traditional somniferum. And the biggest advantage of our variety, it does not have the morphine pathway. And also it has different alkaloids that can be of major potential to cancer research, to pregnant women. There's a lot of different ways that we can use these medications. So I want to touch base on the receptor activity. Everyone thinks that, when I first told my supervisor, hey, we found something, and he said, oh, maybe wrong, right? So we tested with CSIRO, and then we found the compound is real. The thing that separates us from the morphine poppy is morphine actually acts on mu receptors on the brain. So there are 4 receptors in your brain for every one of us sitting here. Mu, kappa, delta. Mu, if a compound acts on the mu receptors of your brain, you get addiction. But our poppy acts on the kappa and delta receptors. And I'll explain to you in a minute. Am I out of time or? All right. So this is our proof of concept, right? We have to prove this to regulators, to Victorian government in Melbourne, and we have a growing facility of 40 acres of land in Melbourne, and we have scientists working on this. We have to prove this every year. And I always joke to my wife that I'm the most audited person because every year I need to do national police checks, FBI, DEA, and all that stuff. Because we are dealing with the Schedule 1 substance here. So this is how we get there. Gene silencing, we silence the genes responsible for morphine pathway. And the second, we found out the cold treatment. Vernalization is a term referring to the cold requirement of a crop. And that's what it needs during that alkaloid pathway to have a thebaine as a main alkaloid. And we also alter the genes responsible for DAHP synthase and also other shikimase pathways. This is the proof that we showed to a lot of people to get our license. And we also alter the ethanol, enol processing things during the extraction process. That's how we get there. I don't want to bore you with a lot of jargon there, but this is the simplified version of, to my right-hand side, left-hand side. If you can see, you can change the color, you can change the disease resistance in poppies. Not in poppies, you can do it in many crops, right? By using CRISPR, if you can change the genes, if you can locate the genes that are responsible, or you can target the genes responsible for, susceptible to disease, you can make a disease-resistant varieties. This is the extension of our work. We also alter some of the tissue transferases that happen during flowering stages of the crop. And we also manipulate the genes out of the pathway which converts reticulin. The biggest question, every time, this is my constant day in, day out. When you say we're working towards a pain relief without addiction, most of the time people say BS, which we got used to it. So now we have to show them how we are different, right? So the opiate pathway acts on your mu receptors, as I told you before. And this is how it acts. So if you see the cortex and ventral thalamus and midbrain and medulla, the opiate activity acts on the neurons, and that's how they inhibit the pain relief, but it also causes addiction. But with the kappa and delta, you have JNK and MAPK signaling happening due to calcium and magnesium enzymes. So the phosphorylation happens pretty quickly, and that's why we are different from a morphine pathway. So in essence, we are not acting on the mu receptors. We are acting on the kappa and delta receptors. That's how we are different from a morphine addictive compound. Again, this graph shows exactly how your pain is actually happening in your brain and why there's a lot of addiction happening. Because when you have a surgery somewhere and a doctor recommends you morphine, you might get addicted a week later. Or if you've been on that drug for 2 weeks, 3 weeks, you don't want to get it off of that pathway because it is, your brain is telling you to take that drug. But with our pathway, it's completely different. And we are doing a lot of research with CRISPR, and in the next 15 months, we have a pathway to do phase 1 clinical trials. We raised good amount of seed money right now, and we are on a pathway to collaborate with NIST and Merck, as I said before, and we are working with a lot of other collaborations. Market opportunity, opiates, it's a billion dollar opportunity. There's hundreds and hundreds of $billion just pouring into morphine. We want to change that. We want to be part of this humanitarian project where you're not causing deaths, you're not causing addiction. We don't want to be a $1,000 drug. We want to be bringing a drug that's available to the market at affordable cost. That's my aim. That's the holy grail of this company. And business plan, we have these milestones. We have a plan for the next 18 months. We're doing all sorts of genomic transfers, knockouts, microarrays with a lot of companies. Right now, we have fully set up a lab in Australia. We also manufacture some of the APIs with a third-party manufacturer, and the goal is to send to Europe, where we have a contracted a partner running that company right now. And these are our licenses. It took years and years just to get here. Collaborations, NIST agreed us to be part of our journey too. These are my publications and some of the references if you want to check all our work. And this is our website, msgene.com. This is us, We're the world's first company trying to get the pain addiction without pain relief without addiction. Thank you for your time.
Thank you so much, Raj. We may have time for one question. If anyone has a question for Raj, he does apologize. He has to rush to a flight, so he will leave before the end of the workshop. So now is your opportunity to ask him a question if you have any. Yes, there's one there.
So thank you for the very nice talk. I hope you succeed. I was just wondering, if you use a different pathway, how strong are these, is this medication in pain relief?
If you use what?
So you use different receptors to cause pain relief, but do you need a higher dose, or is it as strong as when you would use morphine?
That's a great question. If I'm going to answer that, all the clinical trials going to shut down, because that question, it needs a lot of clinical trials to understand what's the dosage, what's the commercial dosage going to be.
No, but I don't mean the dosage, but is it as effective? You could take an aspirin, but that doesn't do
it's
the
not
trick.
effective. We think that at this stage, the low doses are enough. But we are testing on the animal models right now. Once we get the data from that, so we can design what sort of 3 different doses that can be done in humans.
Yeah,
So,
but it has the potential to be as strong as morphine.
yeah, true. But we have no idea until we conduct clinical trials. Yeah.
Thank you.
Thanks.
In the interest of time, we're going to move forward with our next presentation. So thank you so much, Raj.